My name's Dorothy. I'm at Emory School of Medicine, where I've had my lab for just under 3 years. So everything I'm going to be sharing with you today is our unpublished preliminary work trying to investigate paradigms of centrosome regulation. And so to just briefly outline my talk, I'll be introducing what centrosomes are, why we're interested in studying them, our efforts to understand how they're regulated, and a little preview into how we're interpreting our new findings and where we want to go in the future. So to begin, centrosomes are organelles within the cell that function as microtubule organizing centers. And centrosomes themselves consist of what we can consider two main parts. Residing at the core are a central pair of centrioles, and they're shown here in blue, and surrounding the centrioles in orange here is a matrix of many, many proteins called the pericentriolar material. And the job of the pericentriolar material is to dock, nucleate, and organize the microtubules, which are components of the cell's cytoskeleton. So just like our skeleton, the cytoskeleton gives the cell structure and form. In addition, it serves as essentially like a highway track to transmit different cargoes within the cell. And because the microtubules have to physically dock to protein complexes within the PCM, the amount of PCM within a cell instructs essentially the activity or microtubule organizing capacity of a centrosome. So the more PCM you have, the more active the centrosome is, the more microtubules it can nucleate. And that's illustrated here on this recent cover from the JCB journal, where in all of the images, the microtubules are shown in red, centrosomes are shown in green. And you can see with all these mitotic examples on the outside, the centrosomes are relatively large and they're nucleating many, many microtubules. However, this lonely interphase cell in the middle has a much smaller centrosome and it nucleates fewer microtubules. And this is because centrosomes are dynamic organelles. They have to adjust the composition, the amount, and the organization of their PCM so that they can achieve their diversity of functions. In addition to their well-known roles in cell division, where they're required for efficient mitosis and maintaining genome stability, centrosomes function throughout the cell cycle. I mentioned intracellular trafficking of organelles, proteins, and RNAs, which is something we're very interested in. They also, in some cell contexts, can direct the trajectory of motility. And residing at the core are the centrioles, which when they dock to the plasma membrane, become the basal bodies for ciliogenesis and the formation of flagella. As a consequence, when centrosomes are deregulated, if they don't have enough of their PCM or if those components are mutated, the result is a number of human pathologies, including the #1 heritable cause of human microcephaly. Their links to cell division drive genome instability, and centrosome aberrations are very closely correlated to poor cancer prognosis. And polycystic kidney disease is the most common heritable form of human ciliopathy, again, given the centrosome's role in cilia formation. To understand centrosomes and their regulation, we choose a model organism. And our favorite model organism is Drosophila melanogaster. The reason why we love Drosophila is because it has a very simple genome, only 4 chromosomes, yet it bears striking conservation to our own genome. They're also very simple, easy to maintain, they're fast, and we do a lot of imaging in our lab, so we love the fact that so many of their different tissues and cell types are amenable to a variety of imaging techniques, and I'll be showing you examples of several of those today. The system in the Drosophila that we focus on is the early embryo. Now, the Drosophila embryo, if you're not familiar, develops as a syncytium. So what you're seeing here are several different nuclei, all within the context of a shared cytoplasm. And this particular embryo is expressing stably GFP-tagged microtubules. And as the movie plays, you can see that all of these nuclei are dividing in synchrony. And the microtubule organizing centers at each pole maintain microtubules throughout the entire nuclear cycle. So we love the system because these divisions are fast. They're on the order of 10 minutes, and you can image multiple division events all at the same time. And so as embryogenesis proceeds, we're going to go through 14 of these divisions, before cellularization occurs. So we can mark different points in developmental time based on where we are in these nuclear cycles. And centrosomes are absolutely required for embryogenesis to proceed. So as geneticists and developmental biologists, we also live the system because we can study cell biology, but we have clear markers if there's a problem going on. We can tell if the division apparatus is dysfunctional, and there's a readout, typically embryonic lethality or genome instability. And so we and others are very interested in what regulates centrosomes and centrosome activity. And the PCM contains, depending on who you ask, hundreds of proteins. As a field, many of us have focused on a very small cohort of those proteins. But long ignored is the localization of several mRNAs that go to centrosomes. So this has been described in a number of cellular contexts over several decades, actually. But the functional role of these RNAs remains essentially unstudied. And so given our background in RNA localization, we thought this would be an interesting problem to pursue. And so the idea is that RNAs localize to distinct subcellular compartments. And the question is, why would some RNAs localize near spindle poles? So these are images taken from Eric Lecuyer, who maintains an active database. So if you're interested in RNA localization in Drosophila, I encourage you to visit his website. You can look up your favorite RNA molecule. And the RNAs I'm pointing out here are some of the ones that we're studying in our lab. And we're choosing to focus on RNAs that encode known centrosome regulators. The idea being that we know that these gene products influence centrosome activity or spindle alignment. So the question is, why is there RNA there? Could there be some link perhaps to local translation or giving structural stability to the centrosome structure? So the concept of RNA localization is highly conserved from bacteria, yeast, up to humans. And very well studied, for example, in neuronal contexts where neuronal stimulation can trigger local translation of mRNAs located very, very far away from the cell body. And elegant studies in the oocytes and embryos of a number of model organisms have illuminated fundamental principles of mRNA localization and their key role, for example, in patterning and giving rise to differential cell fates. But RNA localization, what a lot of people maybe don't appreciate, it also functions in acute cellular contexts, maintaining cellular adhesions, influencing cell migration. So it's a fundamental paradigm that essentially compartmentalizes cells and gives rise to discrete protein domains. And the way this happens is typically RNA localization is coupled to translational control. And so this makes the process very efficient because individual RNAs, of course, can be translated multiple times. So it's more efficient for a cell to localize a couple RNAs than it is every protein molecule. And you might consider that not every protein needs to be exposed to the entire cellular milieu, and so it limits exposure of certain components that you might want to have turned on at a specific place at a specific time. And so how are we addressing the question of RNA localization? Well, as I mentioned, we're interested in studying RNA specifically at the centrosome, which is a very discrete organelle within the embryo. What I'm showing you here is structured illumination microscopy of a centrosome taken from one of those embryos that I was showing you from the movie. This donut that you see in the middle is the centriole, and the yellow arms extending is pericentriolar material, or PCM. And so this entire structure has a diameter of about a micron, which is pretty small. So how are we going to resolve RNA localized to these discrete structures? More importantly, we want to be able to quantify that RNA so that we can tell changes over time or at different developmental points. And so the method that we're using is called single molecule FISH. The way this works is you take about 20 base pair oligos and you tile them across your coding sequence. And each oligo has a fluorescent tag on there. And if you have 10 or more of these oligos, that's sufficient to generate a point of light that you can detect on a standard microscope. And so this is for me to remind you that everything I'm going to be sharing today is work from one of our postdocs. This is Pearl, who's been really spearheading this project, investigating RNA localization. And so the first thing we needed to do is really assess if the RNAs that we were interested in localized to centrosomes and if they did so specifically. And so this is an image of one of our control RNAs. GAPDH is a metabolic enzyme. It's expressed very highly. Each of these dots that you see here is a single mRNA molecule. And even though it's very highly expressed throughout the embryo, the centrosomes shown in green do not show any particular enrichment of this mRNA. However, when we look at one of our candidate mRNAs, centrocortin, we notice something quite different. Now you see these clouds of mRNA clustering near the centrosomes, and here are the insets zoomed in for you on the bottom. So we became very interested in following up on this, and so we wanted to determine quantitative ways to define localization. And that's really the best advantage of single molecule fish. You have resolution, but you can quantify it very easily. By simply segmenting your image, you can determine where your cell boundary is, where your area of interest, in our case the centrosome, lies, and then you can threshold your objects, which in our case is the RNA, and calculate different parameters. This is an example showing you if you wanted to calculate the fold enrichment of all the mRNA that's at the centrosome. And so Pearl's been working on a number of different methods of quantifying this data, and what I'm showing you here is an example where if we compare localization early in embryogenesis to a little bit later, 3 cell cycles later, we see this great increase in the amount of centrocortin mRNA at the centrosome. And I want to emphasize how conservative this measure is, because we're talking about percent of RNA within 1 micron. So this is essentially in the PCM of the centrosome itself. It already starts out very significantly enriched early on, but then it really ramps up as development proceeds. So what we've identified is both a developmental and a cell cycle dependent enrichment of this RNA as centrosomes, suggesting that this is somehow a regulated process. So what is centrocortin? Centrocortin was originally identified by our colleague Tim McGraw as an interacting protein with another major centrosome protein, and mutants that lack centrocortin protein form these very dysfunctional spindles. So this is a control embryo on the right, zoomed out and then zoomed in, and you can see these beautiful mitotic figures. That's the normal case. But in a centrocortin mutant, if we zoom in, we have these multipolar spindles and fused spindles, which are major contributors of aneuploidy and embryonic lethality. In humans, centrocortin is related to 2 related gene products. This is Tim's alignment showing you the level of conservation between these genes. And in humans, mutation is associated with a neurodegenerative disorder. But surprisingly, actually very little is known about the normal function of this gene product in humans. So we decided we're going to follow up on centrocortin. And we were very interested in the dynamics of its localization pattern, because what we saw was initially the RNA seemed to be radiating somewhat symmetrically from the centrosome, but over time it started to form these higher order granules, which we didn't see in our other mRNAs. So this was something of a unique feature to centrocortin, where you have just these masses of RNA ribonucleoprotein complexes accumulating near centrosomes. So Pearl calculated the incidence of these granules over the entire course of early development, and she finds that at some point, essentially 50% of all of the RNA is within these higher order structures. So of course, RNAs are regulated by RNA binding proteins, and granules are usually sites of some kind of RNA processing or RNA metabolism. So we wanted to know what was in these granules so we could begin to define their function. We were very interested to see that centrocortin granules not only contain their RNA, but they also contain centrocortin protein. And I'm just showing you here a quantification indicating the high level of colocalization. So then we wanted to dive in and see, well, what other kind of RNA binding proteins would we identify in these granules? Because there are some RNA binding proteins that preferentially will localize to one flavor of granule than another. And one of the proteins that we looked at was FMR1. So FMR is associated with Fragile X syndrome, which is the #1 cause of human intellectual disability. It functions as an RNA binding protein. It's a translational repressor. It's often found in stress granules. So we did a number of assays with FMR. What I'm showing you here is a PLA, or proximity ligation assay. This is essentially a form of colocalization where you only detect incidence of colocalization within 40 nanometers of each other. And so it registers as single points of light that you can then quantify. And what we see is a significant number of PLA foci when we look at centrocortin protein and FMR1 protein. We wanted to verify this biochemically, so we did an immunoprecipitation experiment. What you're seeing here in lane 1 is just the input centrocortin protein and FMR1 protein. These are 2 negative controls in lanes 2 and 3. And then lane 4 is where we're actually doing the IPs. So centrocortin can IP itself, and it also IPs FMR protein. Of course, what we really want is functional data to show that FMR is doing something. So what we did is we turned to genetics. So remember, I told you that centrocortin starts out at the centrosome, but over developmental time becomes more and more accumulated at the centrosome and forms these granules. So if we look at an early developmental stage, centrocortin looks a little more diffuse. We don't see granules. However, in the FMR mutant, now we have precocious recruitment of centrocortin and precocious formation of these granule structures, suggesting that FMR functions to limit the formation of the centrocortin granules. I also told you that FMR functions as a translational repressor. So one model could be that FMR might influence the translation state of centrocortin itself. And to assay that, we performed a quantitative Western blot where I'm showing you a control on the left where centrocortin protein is on the top lane. And when we delete FMR, we do see a significant increase in centrocortin protein. And so we're beginning to address our overriding question that we have, which is why are RNAs at the centrosome? We envision several potential models, which we do not think are mutually exclusive. And we think that probably different RNAs are at the centrosome for different reasons. In the case of centrocortin, it seems like we've identified a potential role for translation regulation. And our model, which we are currently testing in the lab, is that the Cen granules are active sites of Cen translation. And we're using tools that will allow us to directly test this. And we also further hypothesize that FMR1 limits the formation of these granules and the translation of centrocortin protein. And again, we're following up on this with some genetic assays. And to more directly assess the role of RNA functionally locally at the centrosome, we want to play around with mislocalizing the RNA. And one of the ways we want to do that is by manipulating RNA localization. So RNA typically localizes through elements within its 3' untranslated region, or 3' UTR. These can be sequence or structural dependent, and these are often recognized by these same RNA binding proteins that I was referring to. These are the so-called zip codes. And there are 3' UTRs that have very strong localization elements. And one of these is bicoid, which is the anterior morphogen in Drosophila embryos. And by simply swapping the 3' UTR, we can ectopically localize centrocortin to the anterior pole. So instead of normally being distributed throughout the embryo and enriched at centrosomes, the expectation is that centrocortin protein would no longer be localized to the centrosome. And we would like to see what the functional output of that situation would be. And that is something that we're working on now. We're building this fly. And so the take-home messages from today is that RNA is localized to centrosomes, and we're trying to figure out what they're doing. And we think we've identified a really nice model system that will allow us to address this question using a number of complementary approaches. And specifically, we've identified a novel RNP, and we're trying to figure out what that RNP is doing. We think it's a site of translational control. And we're looking forward to the next push going forward to really identify what these RNAs are doing, which has been a long-standing question in our field. And so I just want to thank members of my lab. And again, Pearl is the one who's done all of the data that I've shown you today. Our lab is young, but it's growing, and we have a number of undergraduate researchers too who have been contributing to their own projects, which I look forward to presenting in the future. And I'm happy to take any questions if there's time for questions. Thank you. Yes, I will. Yes.
So I assume that you know that Cen is a regulator of the centrosome through genetics, right? You delete Cen or you downregulate it, right?
So the question is regarding centrocortin and whether the functional aspect of Cen is its protein or its RNA. And thus far, the only work that's been done is what I've presented to you, and then our colleague Tim McGraw, who identified centrocortin. And so he has a beautiful paper in Current Biology where they looked at centrocortin mutants and they looked at what centrocortin binds to. So the protein does seem functional, but he didn't look at the RNA because he didn't have a reason to. And so that's part of what we're trying to do, is to separate out the RNA localization to see what happens. We don't know if centrocortin protein will be made at this new site or not, but that's why we're trying to manipulate the localization to uncouple. Mm-hmm. Yes.
Are there any already described centrocortin mutants for Drosophila?
Yes, so the question is, are there mutants that we can use to study centrocortin? And there are. And so we are using the same mutant that the McGraw lab developed, and they are homozygous viable, and they have a low incidence of embryonic lethality, however. There's a question in the back, yes.
I was just curious, so you're making a big way model to relocalize the RNA. I was wondering, have you seen these same types of polarizations of these RNAs in the oocyte? Because that's when Bigway is being localized, right?
Right. So the question is, if I can paraphrase, when does the localization begin? Because yes, it's true that most of these localization patterns are established in the oocyte. So this is something that I'm very interested in looking at. We have the published work from the screen looking at embryonic localization patterns, but we don't know what happens in the earlier embryogenic stages before all these rapid syncytial divisions where all the nuclei and the centrosomes are at the surface, so they're very easy to image. We don't know what happens earlier, and we don't know what happens in the oocyte. This is something that I keep trying to prod people to look at.
Well, my follow-up question then is, do any of these mutants have like a maternal effect?
Yes, essentially all of them. So the question was if there are maternal effects. So as I mentioned, centrosomes are absolutely required for embryogenesis, and the way we even generate these animals is through mutant mothers. We're using embryos from the mutant mothers. Yes. Is there a question?
I just had a question about ribosome enrichment. Is there ribosome enrichment at the centrosomes?
Yes.
Or are ribosomes just exactly first?
Right. So the question is about ribosomes, which if we envision a model where translation might be happening or translation regulation, then are there ribosomes there? So we and others have looked, so in other models where centrosomes, or where RNA localizes to centrosomes, yes, people have identified different translational regulatory proteins and ribosome components, and we have started to look at this in the embryo. We are looking at the small ribosome subunit. With the RNA binding proteins and with the ribosome components, they are everywhere. They're not excluded from the centrosome, and that's part of the reason why we went to this PLA assay, because I could show you co-localization with FMR, but it's everywhere. I wouldn't be convinced by that data. The PLA is a lot more high resolution, it's quantitative. And so what we're working on now in order to visualize translation is a pure PLA. So we can actually look at actively translating ribosomes where they are.
What is the half-life of this mRNA, the Cen?
So we don't know what the half-life is. So through other data, we are thinking that there might be degradation of the RNA throughout embryogenesis. So we know that after cellularization, we have the maternal to zygotic transition where essentially all or 70% of all the maternal load of RNAs get degraded, and then zygotic transcription starts. That's normal for most RNAs, but other RNAs have their own half-life. And so what we're doing now is doing timed narrower windows to define quantitatively how much RNA is in early versus late. We have some of this data just from the SM fish experiments themselves. We don't notice any rapid decay of the RNA, but what we want to do is quantitatively compare the wild type to the FMR, so that's why we're going to the qPCR.
So I'm going to ask you an entirely different kind of question. Yes. So I teach intro bio for majors, yes. And one of the hardest things for me to figure out what to teach is what to teach about cell biology, like especially cell division and cell association. And so given what you know, what do you think is actually important for them to learn coming into undergrads?
Oh, so yeah.
PMAT.
Yes,
right?
yes. The question is regarding an intro bio class. how do you choose, essentially, out of a huge textbook of material and a huge body of literature, what to present in terms of cell biology? And I mean, I don't know that I'm any authority to say this. I think the goal is to get people excited about what's going on. I think cell division is fundamental to development, to us, to disease. So cell division seems like a pretty important aspect of cell biology to study. And people love movies. So it's a dynamic process that's well studied and easy to compare in different cellular contexts and models. So I think I would go with cell division. I think that's very important. Yes.
Speaking of videos, you showed us a video of this succession of microtubules. Is that your video?
Yes.
Everybody in this room is like, ooh. Can you share that video with us?
Yes,
because I would love to use that in my class.
Yeah, absolutely. Happy to share that. Yes, yes. That's why we love the embryo. It's really great for imaging. Yes. Great. Thank you.